This conference will now be recorded.
Hello, class. So we are picking up where we left off with the nervous system, and I'm just going to do a really quick recap for you of what we've learned so far. I've included little summary videos. There's 3 parts so far that I've posted here that you can, basically copy and paste this into your browser. Check out a 10-minute video. It's more information than you need to know, but it kind of puts things into context, perspective. It's entertaining. It's optional. You won't be tested on anything from the video. Yes, so we were talking about the nervous system and how it has really 3 different functional organizations: a sensory function that really picks up stimulus from the environment, or even internally. Internal environment, okay, so I'm just using that as a broad general term as environment. There's an integrative function where some processing of sensory information is happening to make a decision about what the appropriate motor response should be. And motor function is where there's a command from the central nervous system to activate, another muscle, gland, or organ. And divided into 2 major divisions, including the central nervous system, which is the brain and spinal cord, and then everything else, which is the peripheral nervous system. We went over basic anatomy of neurons, how they have dendrites that pick up information from other, well, they could have sensory organs attached to them, which would pick up information from the environment, whether that be the skin or organ, joint, right? And then it's connected to a cell body, which then has a axon, and at the very end is the axon terminals with the synaptic end bulbs. So we're going to be focusing some more attention on this area today. We talked about different kinds of neurons. And there's those 3 different kind of functional types: the sensory neurons, motor neurons, and interneurons. We talk more about this today. I introduced you to neuroglial cells, which are not conducting or electrical conducting cells. They are supportive cells of the nervous system. We talked about the membrane coverings of the nerves and how that's organized. I introduced you to the cranial nerves. And we talked a little bit about Bell's palsy, which is a condition with facial nerve. Spinal nerves, the 31 pairs, and how those are properly named and numbered. And then we went over the 4 major plexuses of the body: cervical, brachial, lumbar, and sacral. So you can review that. Okay, so this brings us to where we are at today. We're focusing on action potentials and what happens at a synapse. This is a more technical part of the chapter. A lot of students get a little bit confused here, so I've done my best to make it as simple as possible, and I think I did a good job. I don't know, we'll see what we end up with here. But of course, if you have questions, like, bring those to me. Your textbook puts it in a very simple way that you can understand. So yeah, let's just get right into it. Again, there's a crash course short little video there about action potentials that you can take a look at. So impulse conduction, this is a quote from your textbook. Impulses are created by changes in the environment that stimulate dendrites, initiating a sequence of electrical events that travel along a neuron's plasma membrane all the way down an axon. Okay. When an impulse reaches the axon terminals, it causes the release of neurotransmitters that build a chemical bridge between one neuron and another neuron or effector cell. So that's a summary of what we're going to be learning today. So here's 2 neurons. Well, actually, technically 3 neurons, but 1 full neuron in the middle. We have the axon terminals of one axon synapsing with the dendrites of another neuron, transmitting an impulse or action potential along the axon until it reaches the axon terminal, where it then synapses again on another dendrite of a neuron. We're going to talk about what exactly happens at the plasma membrane level, all the way down the axon, and then what happens at the axon terminal with neurotransmitters and the synapse and all of those things. So what do you know already? So last in basic year, you learned about the neuromuscular junction, where the nervous system, the motor neuron, tells muscles to contract, okay? And that was stimulated by an electrical signal from the nervous system. And this is what you know now, right? So here we would have a sensory neuron in blue, a interneuron in purple, and a motor neuron in red. The sensory neuron has these receptors at the end, or free nerve endings, or they may have an actual a sensory apparatus at the very end. So in this case, somebody is sensing something sharp poking into their skin. And that message is transmitted to the central nervous system at the spinal cord, which is what this is, a segment of the spinal cord, and relays that information to an interneuron, which then synapses on another motor neuron, telling the muscle to contract, likely to, I don't know, try to get the sharp item out of the skin. You also knew that an axon of a motor neuron had many different terminals that synapsed on different muscle fibers and caused coordinated muscle contraction. All right, so I just wanted to kind of build on what you already know. Same thing, except the examples that we're going to talk about today, I mean, you can have the effector being a muscle gland, right? Exactly like that. But we're also going to talk about how axons communicate with each other, or neurons communicate with each other. We also had the example of the synapse, where the synaptic end bulb of a neuron connected, well, I mean, very close proximity to the motor end plate of a muscle cell. The muscle cell's plasma membrane is called a sarcolemma, right? But an axon is just called a plasma membrane. Okay, it's just the skin or outer covering of a cell. They just have different names depending on where they're found. So yeah, you already know about how synapses work, where a synaptic end bulb is containing these synaptic vesicles that have a neurotransmitter. In a neuromuscular junction, the neurotransmitter was acetylcholine. But in neurons, oh my God, I can't talk. In neurons, there's different neurotransmitters depending, okay, on an inhibitory or an excitatory effect, which can get very complex, but we're keeping it real simple, and I talk about that a little bit later on. Okay, so a couple definitions. Nerve impulse is just an electrochemical signal carried by neurons, and neurotransmitters are a chemical bridge between one neuron and another. Okay, I liked that. It's a chemical signal or chemical communication. So the first thing you need to understand is the resting membrane potential. When a neuron is at rest, the neuron has a very specific charge to the plasma membrane, and it's negative, okay? So at rest, the cell's membrane has a negative charge of minus 70 millivolts. And this is due to an excess of negatively charged ions on the inside of the cell. So here we have a neuron and we have the axon, and you can see the negative inside the axon and the positive in the interstitial fluid surrounding this neuron. And when you zoom in, this is not from your textbook, it was actually from your previous textbook. And luckily, we don't go into too much nitty-gritty details about what kind of channels they are and all of that jazz. But it is important to kind of highlight this. You won't have to, you know, you won't be tested in all the little specific things here, but I want you to understand this concept. So here we have a plasma membrane. You can see the phospholipid bilayer of all cell membranes. And all cell membranes have these integral membrane proteins interjected in them. And they are typically channels to transport ions across the plasma membrane. So the blue part is the interstitial fluid. This is the fluid surrounding a cell. And then the cytosol is the fluid inside of a cell. And the plasma membrane is separating them. The charges between the interstitial fluid and the cytosol are different depending on what components are inside the cell and what kind of components are in the interstitial fluid. Inside the cell, there's more negatively charged ions in the form of phosphate and protein. And on the interstitial side, the outside of the cell, there is more positive charged ions. And so this creates an electrochemical gradient. And nature hates gradients, okay? It always wants everything to be equal and even on either side of the membrane. So because there's a charge that's separated by a membrane, this gives it potential energy. So recall that areas of high concentration want to move to areas of low concentration. So if we have a lot of sodium ions in the interstitial side and few sodium ions on the cytosol side, nature, the laws of nature and the power that be, wants the sodium to come into the cell. But it is not allowed because all of these channels are closed, and so it can't get in there. This is potential energy. Okay, there's some force that wants to bring the sodium in, but it's not allowed because there's no way in. All right. When channels aren't open all the time, they're called gated channels. Just so you might recall that from last year. Okay, threshold stimulus. So when a neuron is at rest and nothing's happening, there's no stimulus, this is what's happening at that resting membrane potential. Okay, all the channels are closed, it's more negative on the inside of the cell, and that's it, minus 70 millivolts. I like the way that looks. Sorry, it just looks really confusing. I'm gonna just fix that real quick. So threshold stimulus, this is where something happens. A stimulus, a change in the environment, causes a channel or all the channels in the plasma membrane to open and allowing the ions to pass across. A threshold stimulus has to be large enough. It has to be strong enough to reach threshold, okay? That's why it's called threshold. Otherwise, it's just a stimulus. Your body might like actually ignore it if it's not strong enough. But once it reaches threshold, this means that there's a large enough shift in the ionic charge of the plasma membrane to make something happen. And remember, we started off at that minus 70 millivolts at resting state. Well, a threshold stimulus has to cause enough sodium channels to open to allow sodium into the cell to create this a little bit more positivity on the inside of the cell. Okay, so whenever there's a stimulus, it'll open some sodium channels, and then those sodium ions are allowed to come across. And this cytosol side gets slightly more positive, reaching a minus 55, right? Minus 55 is more positive than minus 70. And then this is reached threshold. And once you have a threshold stimulus, then you can generate an action potential or impulse along the axon. the neuron generates this action potential at the axon hillock. The axon hillock is where the cell body meets the axon, and two phases occur at this point. Okay, so for an action potential, there's the depolarization phase and a repolarization phase. Now, these words can get confusing because you have to kind of remember this. Depolarization is making the cell membrane more positive, and repolarization is bringing it back to negative. Ready? Depolarization makes it more positive. Repolarization brings it back to negative. So like minus 70 at the resting state. So here's just a little visual of that. So here we have that minus 70. This is that resting state, resting membrane potential. And then you have your threshold of minus 55. And unless the stimulus is strong enough to reach that minus 55, like if it's not, nothing will happen. But once it reaches that threshold amount, then you have depolarization occur. And this creates a much more, well, what happens in the depolarization phase is that the gated channels open in the neuron's plasma membrane that allows the positive sodium ions to diffuse into the cell from the outside, okay, making that section or segment more positive. Just to recall here the difference between diffusion and active transport. Remember how I was talking about potential energy? Well, if it is moving along its gradient, its concentration gradient, where there's less sodium inside the cell than there is outside, so the sodium outside is just naturally going to come in. This is diffusion. This is without adding any extra energy to it, right? Potential energy, it can do work. So that I just wanted to kind of clear that up and make sure that you're understanding the difference between diffusion and active transport. I can't imagine myself testing you on anything very like complex like that, but it can be a place where you get confused, like why this happens. So hopefully I didn't confuse you there, but just know that because of the difference in that concentration gradient, sodium wants to come into the cell without ATP. It's just using its gradient to move in. So this causes the inside of the cell to become more positive at this region, right? You have the stimulus, it reaches threshold, all of the sodium ions rush in through the channels, and this section is more positive compared to its adjacent region. At repolarization, this is where potassium channels are opened and potassium starts to flow out of the cell, reestablishing that negative resting membrane potential. It overshoots a little bit, so there's a little bit of a hyperpolarization phase right afterwards, but eventually it just regains its homeostasis and ends up at that minus 70 resting membrane potential. It happens quickly right after the depolarization. Positive potassium ions, yes, flow out of the neuron, returning to resting. And there's a little bit of a refractory period. It's a very short period of time after the neuron has depolarized. Depolarized, repolarized, and then it kind of has this refractory period. Not until the neuron has repolarized can it respond to another stimulus. So it's kind of like your textbook gives a really cool example. I actually like their examples of when you're using a motion sensor paper towel dispenser. If you grab it and it's like it doesn't see you the second time you go and grab a paper towel, you have to kind of wait until it resets. That's exactly what the refractory period is. So how does an action potential move along the axon? This is called propagation or conduction. Those words can be used interchangeably. I think propagation is what your textbook kind of prefers. So the opening of sodium channels and an influx of those sodium channels coming inside the cell across the plasma membrane during that depolarization phase causes the adjacent portion of the membrane to reach threshold and open more sodium channels next to it. This is a domino effect all along the axon until it finally reaches the very end at the axon terminal. Saltatory conduction is a name for how myelinated axons potentiate the action potential along the axon. It's a much faster process because remember, in myelinated axons, the Schwann cells create these myelin sheaths that cover segments of the axon. And then you're left with these little bare segments called the nodes of Ranvier, which are gaps between those Schwann cells, and potassium and sodium channels are located in these regions. So you get a stimulus at one end and you have that depolarization, repolarization, and it's propagating all along the axon, except it leapfrogs from node to node. And that's why you get a much faster propagation. This is called or termed saltatory conduction. So that's it. Action potentials in a nutshell. You've learned it. So I hope that you are able to understand what we've talked about there. Of course, we can go over it and we will definitely be doing that in our review class. The last part of what we're learning today is about synapses and reflexes. So here's another little video that you can watch about synapses. So the synapse is a junction where the electrical signal passes from one cell to another. Neurotransmitters cross the synaptic cleft here, and different neurotransmitters such as acetylcholine, norepinephrine, dopamine, serotonin, endorphins, there's many more, can be transmitted to the next cell, the next axon. I'm sorry, not axon, neuron. And they can have an excitatory or inhibitory effect. All I want you to really know is about this is that the vesicles release the neurotransmitter into the synaptic cleft, where the neurotransmitter will join a receptor on a protein on the opposite side of the synaptic cleft. Okay, and we term that the postsynaptic neuron. So the neurotransmitter is joining on a receptor of a protein. So an excitatory neurotransmitter, the presence of an excitatory neurotransmitter will create a threshold stimulus and depolarization. So it continues transmitting the message. It brings it closer to threshold, that -55, and then the action potential can continue along that neuron as well. An inhibitory neurotransmitter will prevent the receiving neuron from reaching threshold, right? So it'll make it more negative and will it'll prevent generating the action potential. Okay, so neuronal pathways are just one-way routes from one region to the next, and they're termed afferent or efferent. Afferent and sensory can be used interchangeably, and efferent and motor can be used interchangeably. In physiology, afferent, you always remember arrive. Okay, afferent, arrive, efferent is exit. And you're thinking about it in terms of the spinal cord. So a sensory neuron is going to arrive to the spinal cord or the central nervous system. And a motor neuron or an efferent pathway is going to pull the impulses away from the central nervous system. Simple, right? We learn more about the spinal cord and your peripheral nervous system later on, but yeah, as long as you just understand that this it's a one-way transmission. The sensory goes one way, the motor goes the other way. So a reflex arc is a very simple, because obviously like higher level processes require far more neurons than a reflex. So like thinking and remembering and integrating information requires so many more interneurons. Okay, so that's where things get complex, is this the purple interneuron, okay, can be like very numerous and actually transmit a signal from one stimulus to many different locations inside that central nervous system, different areas of the brain and spinal cord, and to make a more complex decision about how that that impulse should be distributed along the efferent pathway. So reflex arcs are very simple, and all that you'll have to be able to answer questions about on a test. They provide a predictable motor outcome for a specific sensory stimulation. And the two classic examples are the deep tendon reflex, or a stretch reflex, and a withdrawal reflex. Deep tendon reflex is where the examiner will briskly tap the tendon of the quadriceps and give a very predictable knee extension response. The deep tendon reflex requires only two neurons. Okay, that's important to remember, two neurons. One is a sensory, detecting that stretch of the tap in the tendon. And it creates a synapse between a sensory and a motor neuron. And the motor neuron brings information to tell the quadriceps to contract, and therefore you get that knee extension movement. In a withdrawal reflex, this would happen when an individual senses something that's hot or sharp or harmful in some way, and the reflex is to withdraw, right? So this person, I think, is touching a nail or a hot stove or whatever you want to have that stimulus be, and the decision is made at this cord level. So it's not requiring that your brain get involved in this and say, oh my goodness, that feels hot, I should take my hand away, right? It's trying to protect the organism, right? And if it's detecting something that's causing tissue damage, the immediate reaction is to pull away before they even understand what's happened. There is an interneuron here that integrates between the sensory and the motor neuron in a withdrawal reflex. And that interneuron can actually convey messages to the rest of the body as well, the brain, like in the brain, I should say, so that the person can, have a response to it. And that's unique. That can be really unique. So some people can burn their hand and interpret that as like an emotional like insult, right? You can kind of see that in kids where they get hurt and they're like, ah, but then all of a sudden, there's a stronger emotion with them, like, like what's happened to me? I'm so, like kind of freaked out. Right? Or you might, or it could be like, oh, I did it again, right? You have these memories and these interneurons, like they don't even really understand how this happens. But that's what interneurons do, is it transmits this information to different areas of the body, and you can have many networks, which we'll see in just a minute. But anyways, that's what happens in a withdrawal reflex. There is one interneuron in a withdrawal reflex, no interneuron in a deep tendon reflex, and they're all handled, reflexes are all handled at the spinal cord level. It's a predictable motor response to a sensory stimulus. Okay, so neuronal pools and circuits. Neuronal pools are defined as billions of interneurons, those are those purple neurons, organized into functional pools. Yeah, and this is just to organize information, and that's really all you have to know about that. Circuits are neural networks of sensory, associative, and motor neurons. I guess I should have called this interneurons. Interneurons and associative neurons are exactly the same. Okay, don't get confused by that. But these circuits are going to have networks that are convergent or divergent. Voluntary and involuntary control is convergent circuits, where divergent circuits are creating systemic effects. Remember, systemic effects are, an example is like giving somebody a massage on their neck, causing their blood pressure to lower, for them to feel a sense of well-being, to have like also their facial muscles relax, you know, these systemic effects in the body. So your divergent circuit is a circuit in which an impulse from one neuron spreads out the information to several other neurons, so it can have that systemic effect, right? Like it can bring that information to many different areas of the body. Where a convergent circuit is when an impulse from several neurons converge onto a single neuron. And then finally, just a pathology alert. Obviously in pathology, we talk a lot more about the nervous system, but just to make this connection, when you have pathology in the nervous system due to compression or tension syndromes, these are syndromes such as sciatica, thoracic outlet syndrome, and carpal tunnel syndrome. Sciatica, actually, I would say this is more appropriately named piriformis syndrome, because you have this peripheral nerve, which is the sciatic nerve, being compressed under a tight piriformis muscle. And then signs and symptoms of neural compression involve numbness, tingling, so like pins and needles feeling, muscle weakness. People will tend to, well, if it's happening in their hands or arms, they'll tend to drop things or feel numb in their fingertips. They can't feel things properly, so there's just like not accurate coordination between the muscles and the nervous system. And also chronic aching and pain is also associated with nerve compression. A manual therapist can be a godsend for these types of things, because you can relax muscle or reduce the tension on tight fascia, then the compression on the nerves is relieved and the symptoms will go away. There's also potential that there could be a more severe neurological cause to their symptoms, so you always want to have that in mind. Make sure that they're assessed properly by their doctor. That might include like imaging, things at the spinal cord, because there could be like spinal stenosis that's contributing to the pain and happening more upstream, or even herniated discs and sciatic irritation, right? Alrighty, so that's it for our class today. You have associated workbook pages to go with this lecture. They are pages, it's just one page. Page 101 has a workbook assignment. It's not due, it's for your own learning, and I'm happy to answer any questions that you have. I released an assignment to you all that encompasses everything in the nervous system physiology that we are doing up until holiday break. It's not due until December 22nd, so you still have lots of time. If you're looking at it and you don't know what the answers are, my goodness, you have so much time, you haven't learned everything yet. So don't worry too much about that, and answer it as we, as we get to those areas. Well, that's it for me. I look forward to speaking with you all at our review session coming up, not until October 22nd. And we'll be meeting online at that point in the evening, October 22nd, to do a review for physiology. Alrighty, have a wonderful day. I will talk to you soon. Bye. Bye for now.
This conference will now be recorded. This conference will now be recorded.